Hello everyone, my name is Ananya Chowdhury and today I'm excited to take you on a journey through my summer research project. It's a story about tiny organisms, cutting-edge science, and a molecule called spermidine. Today we'll explore its effects on aging and how it could impact male Caenorhabditis elegans, or C. elegans for short. How it all began. My journey into aging research started in a year-long project under the Duck's Rise Fellowship, where I explored how chronic stress might accelerate aging in college students. My mentor, Dr. Patrick Phillips, recommended a fascinating book called Lifespan: Why We Age and Why We Don't Have to, by Dr. David Sinclair. David Andrew Sinclair is an Australian-American biologist and academic known for his research and controversial claims on aging and epigenetics. He's a tenured professor in the Department of Genetics at the Paul F. Glenn Center for Biology of Aging Research at Harvard Medical School. Intrigued by his work, I learned about a company that he co-founded called Tally Health, which develops vitality supplements. These supplements claim to reverse biological aging by targeting specific epigenetic hallmarks of aging. This curiosity is what brought me here, to investigate one of those key components, which is spermidine. Let's talk about the pill. In Tally Health's Vitality Supplement, each ingredient is carefully chosen for its unique role in promoting health and longevity. First, we have calcium alpha-ketoglutarate, which helps boost cellular energy production and enhances exercise performance. Next, quercetin supports a healthy immune system and aids in clearing out dysfunctional proteins. Then, there's transresveratrol, which is known for activating repair processes and maintaining the epigenome, which is crucial for genetic stability. Fisetin plays a key role in removing senescent cells, which are damaged cells that no longer function properly, which further boosts immunity. However, the compound that caught my interest was spermidine. It's a fascinating molecule with dual benefits. It induces autophagy and enhances mitochondrial function, which ensures that our cells' powerhouses remain efficient. Among all these, spermidine stands out as the focus of my research for its unique impact on aging. So what is spermidine? Spermidine is a natural polyamine that's found in all living organisms. It promotes cellular health by inducing autophagy, which is a process where cells recycle damaged components to maintain balance and function. It achieves this by creating conditions that favor autophagy. It inhibits a protein called EP300 and boosts the expression of key autophagy-related genes. Spermidine also activates transcription factors like TFEB, which regulate the production of proteins involved in this recycling process. By reducing the levels of acetyl-CoA, which is a molecule linked to cellular aging, spermidine minimizes harmful chemical modifications in cells. These processes clear out damaged components and help cells function more efficiently, which scientists believe contributes to longevity. However, there's a twist. The effects of anti-aging compounds can sometimes differ based on biological sex, which is a severely understudied part of drug testing. Here's an example from a mouse study done by Harrison et al. In 2014. When fed an anti-aging compound called acarbose, male mice lived significantly longer. The effects on females were minimal. This raises a big question. Could spermidine also have sex-specific effects? So we arrive at the central question of my research. Does spermidine affect longevity differently in male versus hermaphroditic organisms? Specifically, can it extend the health span and lifespan of male C. elegans? But first, why use C. elegans? C. elegans is an ideal model organism for aging research because of 4 key features. First, they have a very short lifespan of just 2 to 3 weeks, which allows us to quickly observe the effects of interventions like spermidine. Second, their bodies are transparent, making it easy to study their internal structures under a microscope. Third, They produce a large number of offspring, providing plenty of data for reliable experiments. Lastly, their genetics are incredibly well studied, offering a simple yet powerful system to understand how specific genes and pathways influence aging. These traits make C. elegans both practical and invaluable to studying the biology of lifespan and health span. Sexual dimorphism is defined as the biological difference between sexes and is an important consideration when testing chemicals. C. elegans population include hermaphrodites, which are XX. The hermaphrodites have both sperms and eggs, and they also have males, which are much rarer and only produce sperm. They differ in their physiology and in their responses to environmental factors. For example, this study by Liggett et al. Conducted in 2015 showed that glucose significantly affected the lifespan of hermaphrodites, but not males. So understanding these differences is key to interpreting the effects of spermidine on general populations of organisms. A previous study done by Eisenberg et al. In 2009 that only looked at hermaphrodites has shown that spermidine increases the lifespan of hermaphroditic C. elegans by about 15%. But this leaves a gap in our understanding. Does it have the same effect, or any effect at all, on males? This led to my specific focus. Can spermidine extend both health span and lifespan in male C. elegans? To investigate, I designed 3 assays to measure lifespan, reproductive ability, and oxidative stress resistance. These assays are simple yet effective. A lifespan assay tracks how long the worms live. A mating assay tests their reproductive health, since fertility is often a measure of vitality. And finally, a paraquat sensitivity assay measures how well the worms resist oxidative stress, which is a common contributor to aging. To investigate the effects of spermidine on male C. elegans lifespan, I used a protocol modified from the CITP assay. The CITP is the Caenorhabditis Intervention Program, testing program, and they have standardized assays for all types of testing, and I used one of their assays and modified it to fit my experiment. The process began by synchronizing the worms. I transferred 5 gravid young adult hermaphrodites to a plate and allowed them to lay eggs overnight. This ensured that we had synchronized populations starting at the same developmental stage, with day 0 defined as the egg-laying day. Once the eggs hatched, I transferred 40 day 1 adults per condition, spermidine-treated and water-treated, to their respective plates in triplicate for a total of 120 worms per group, which is 240 worms in total. Plates were incubated at 20 degrees Celsius and monitored 3 times a week. During scoring, live worms were identified based on movement, while immobile worms were gently prodded to confirm their status. Dead worms were removed, and lost worms were recorded as missing and censored. This assay allowed us to meticulously track the survival of male C. elegans under spermidine treatment. We did 3 lifespan assays in total. Now, here's where things get interesting. Contrary to what we expected based on previous studies with hermaphrodites, spermidine exposure actually significantly decreased the survival of male C. elegans. As you can see from this curve, males treated with 0.2 millimolar spermidine, which is the purple line, lived shorter lives compared to the control group, which is the black line. This result is particularly surprising given that spermidine increased hermaphrodite lifespan by 15% in the 2009 study that inspired this experiment. These unexpected findings highlight the importance of studying sex-specific responses, as the same treatment can yield drastically different outcomes in males and hermaphrodites. Does spermidine length and lifespan? No. However, let's see if they work for the health span assays. Before discussing the next few assays, it's important to note that fertility in male worms is the indicator that is used for defining vitality, and it decreases naturally with age. The 2013 study by Chatterjee et al. Showed that male fertility drops dramatically from 60% to about 0% in just 7 days. This natural decline provides a baseline for understanding how spermidine might influence reproductive health, because for the mating and the paraquat assays, when we use old worms, we're talking about day 7 worms. The middle-aged worms would be about day 5, and the young worms would be about day 1. So even though males live for up to 20 days to 1 month, they lose their vitality by day 7, which is why day 7 worms are considered old. To test the effects of spermidine on male fertility, I conducted a mating assay. This involved a meticulous preparation to ensure age synchronization and appropriate experimental conditions. On day 1, I set up an egg lay to generate worms for the day 7 stock. Male Hem8 worms were synchronized and transferred to compound plates containing either 0.2 millimolar spermidine or water, which was used as the control. On day 7, I repeated the egg lay process for the day 1 stock and prepared age-synchronized females from the Fog 2 strain. On day 10, the actual assay began. Each plate contained 1 male and 2 females, allowing them to mate at 20 degrees Celsius. After 2 days, I scored the plates for the presence or absence of progeny as a measure of mating success. This process allowed me to evaluate whether spermidine affected male mating efficiency. However, the results showed no significant difference in mating efficiency between spermidine-treated and control males. As shown in the graph, the mean mating success for both groups was comparable at day 1 and day 7. Interestingly, male fertility naturally declined with age, which is consistent with prior studies. These findings suggest that spermidine does not appear to influence male fertility, at least under these experimental conditions. Paraquat sensitivity assay was designed to test whether spermidine exposure enhances oxidative stress resistance of male C. elegans. Paraquat is a plant hormone that induces oxidative stress by generating reactive oxygen species, making it an effective tool for assessing cellular resilience. The process once again involves synchronizing males at different stages: day 1, day 5, and day 7. For each group, Hem8 males were exposed to either 0.2 millimolar spermidine or water on compound plates. On day 10, I transferred 25 worms from each group 20 to 25 worms from each group to plates containing either 4 millimolar paraquat or water as a control. I then monitored their survival daily, transferring worms to fresh plates every 7 days to maintain consistent conditions. This setup allowed me to measure how spermidine-treated males coped with oxidative stress compared to untreated controls. However, the results again showed that spermidine exposure did not significantly enhance oxidative stress resistance in male C. elegans. As we can see from the graphs, the survival times were similar across both spermidine-treated and water-treated groups, regardless of whether worms were exposed for 1, 5, or 7 days prior to the paraquat assay. For example, day 1 worms exposed to paraquat had comparable survival times whether pre-treated with spermidine or not. Similarly, day 7 worms showed only minor, non-significant differences in lifespan between spermidine and control groups. These results align with the other findings from my study, further highlighting the sex-specific nature of spermidine's effects. Unlike hermaphrodites, male C. elegans appear unresponsive to spermidine in terms of stress resistance. So does spermidine lengthen lifespan? No. And health span? Also no. in male C. elegans. However, these findings open the door to several future research directions. First, it's critical to retest spermidine using different concentrations to determine if there's a dose-dependent effect. Second, re-evaluating its effect on hermaphrodites could provide further insights. Lastly, testing spermidine on mutant worm strains could reveal more about the underlying genetic factors that mediate its effects. These studies could deepen our understanding of sex-specific aging mechanisms. I'd like to thank my project mentor, Rose Al-Saadi, and my principal investigator, Dr. Phillips, for their guidance throughout this project. I'm also very grateful to the Phillips Lab for their guidance throughout this project as well. I would also like to thank the CURE team, especially Lynch McCormick, Kevin Hatfield, and JC Bartlett for their support and for funding this research. It was an invaluable experience that allowed me to grow as a scientist, and I look forward to continuing this journey. Thank you all so much for your attention.